I have a few other slides to introduce for the discussion on risk assessment. So last week on Thursday, I was off to dinner with my wife.
Microphone.
All right, microphone. Last week on Thursday, I was off to dinner with my wife, and I was, as I frequently do when I'm with my wife, physically present and mentally absent, thinking about the conversation we would have here. And what I might, oh, don't let that get out, okay? This is, and so when I sat down, they put a coaster in front of me. A coaster. And when the slide comes up, the slide actually helped me get ready for this talk. Fortunately, it's only an hour to go till the end of the session. Speak your mind and ride a fast horse. So not a fast horse, but it's a really short time frame. That's the way I'm looking at this. So this is a quick discussion on how these tools might help me in risk assessment. Okay, me in risk assessment. Each of us really deals with a different piece of the world of how we understand things like mode of action, disease. We take these tools in and then try to apply them. So I think there's some idiosyncratic ideas that we discuss and then the common ones. I've done toxicology, an area which I think has suffered miserably in that time. And I'm going to say we. It's not actually about the field absence of myself. The question is, how much is too much? It's largely what we want to ask ourselves. Chemicals can have an effect. Paracelsus said everything can have an effect at high enough doses. So we want to find out how much is too much in a test system and some way to move that from what we study to people. These are the extrapolations. So my own belief is that almost all the extrapolations we do are consensual. They're based on default methods, and they lack a strong underpinning in biological principles. It's a sorry state of affairs, but it's been due to our ignorance. And in the last 20 years, we actually, and the rules for most of this risk assessment dates back to the 1970s. In the last 20 years, we have a lot more substrate upon which to actually come to conclusions. As a field, we've stepped back and so far haven't had those discussions. We're also moving away from studies we've done in intact animals, systems that will use human cells, or that's the goal. So what might risk assessment look like? What do we have to consider as we go forward? And so I'm going to particularly be thinking about the issue where when we look at 10% of cells responding, it's a dose response problem. When do those 10% respond rather than 20% or 100%? But the idea that you could have them all respond. But the nature of response is quantal. I showed you this before about having networks and then looking at these patterns of response. And this is from John Tyson, and he talked, I'm sorry, but I have to put in pinches. He did a wonderful article called Buzzers and Clickers, oh my, or something like this, where he talked about the underlying biology that gives rise to circuitry characteristics in cells, and has studied the cell cycle, and that's one of his diagrams of cell cycle activity. And it's a parameter estimation problem, and is concluded with the appeal for a greater role of mathematical models in molecular biology. I think there's a greater role for models of how cells respond and how we do risk assessment. Dr. McMullen, asked a question about DNA damage, and it's not, these processes don't just control cell proliferation and cell progression, but networks are involved in how cells respond to stress. DNA damage, oxidative stress, osmotic stress, and a body of work has been done looking at adaptive post-translational control, where DNA damage first actually causes changes in DNA repair centers, and only after the cell is in a position of having insufficient material to make repairs on damage, that it gets into activation of p53 pathways transcriptionally. So there are these tools for looking at this, and my colleague Rebecca Kluhl and Patrick and others have been involved in Schuden in actually both understanding the thresholds that or looking at the data, and that's what's shown in the slide on the picture on the left, where this is micronuclei in response to a particular agent, and it appears to have a threshold statistically, and what's the underlying structure that can give rise to that. I'm not going to go into a lot of detail. Maybe you think I've gone into too much already, but there are two different pathways. One is post-translational, and one is transcriptional. Both of them have a requirement for p53, and the post-translational makes quick repair, and then the transcriptional comes in after damage is sufficiently great that some change in the cell characteristics, a phenotypic change, has to take part. How do we look at this? And this is partly the question. I'm keen on this issue of how we look at cells as an individual unit. This isn't just a question of how we look at cells, but how we look at individual molecules and how we can look at molecules. And these DNA repair centers are formed by different phosphorylated proteins coming together. So to study the process, we have to have tools to look at single cells, and you'd like to have them across multiple cells. So this is the gamma H2AX and p53 binding proteins and how they come together in a single nuclei. The dose response, you increase the dose, you see these form, and at the bottom it just shows you can look at how quickly they form and how quickly they're actually removed. And conditions under which the damage is so great that they're actually persistent. You can't actually resolve them. And because you can't resolve them, you end up having mutations. In this case, measured as micronuclei. This process can be described. And you can populate can be depicted. And you can populate the picture with characteristic rate constants if you can collect data. So it's having single cell responses. In this case, it would be looking at high enough doses, transcriptional activation, which would be clearly, well, I won't say it, because I'm going to get to the question. So, over the morning, in my little two talks, I've actually asked a question about what does it mean to see activation at the cell level with the enzyme inducers. Number two, it's what does it mean when we look at cells in which we're not seeing a change? We haven't seen that activation. So chemical risk assessment has made very little use of this kind of mechanistic information, and especially in dose-response models. So somebody like me, I've spent my whole career doing dose-response models. First, figure out the dose, and then describe the response in terms of the dose. That's it. That's all a toxicology in 20 seconds. And then you have to apply it across multiple different endpoints. But we've had case law. We can argue about it, and the arguments won't do anybody any good. The possibility for me, when I hear about these tools, single cell analysis, being able to look at individual molecules and put them together, is to create another way to look at the problem of when is it too much of a chemical when we actually want to look at it. That's adversity. Okay. Kind of my own personal bias is that most cellular responses are all or none, and we have to actually be able to look at them in more detail. And the networks that control these contain molecular components that have to be described. And I think the comment that was made earlier by Raj is that this idea that you have to have a sense of what's going on and then be able to actually ask how the adversity is embedded there, and then go back to this issue of how would we look at it in terms of the tools we've heard about. Okay, so how do we place the responses of these single cells into a context of those occurring in a tissue or organism? That's one problem. We can get great tools to look at individual cells, three-dimensional cells in culture, but what do they mean with respect to taking these observations back to an individual? That problem's been with us, and I'm not creating a new problem. How do we consider regions? When is the perturbation adverse and when is it not? So I talked about this DNA damage. We can show appearance of DNA repair centers. No mutation, but we've had a perturbation sufficient to cause a change or an activation of processes going on in a cell. Would we in toxicology call that adverse? We can see something. Is it adverse? It's a precursor on the way to adversity, which is mutagenicity, but how do we kind of get that conversation going? The other idea that I hear from, or I take home from this meeting, is this concept that we treat a tissue, we see a movement of cells from one state to another. How does that happen? What causes that change? What propels the change to a new state, and what's the underlying cellular components that affect it? How do we get that to be part of our conversation? Raj brought that up, and we've had several other conversations there. And for me, can a mindset be established to pay as much attention to that network that controls the response as the response itself? We've been quite good at saying all or none. None, basal. All, a response. We want to know the dose response, in my opinion, and also how we actually put together the tools to understand why it occurs. That, for me, would be modern toxicology. And it requires lots of tools, and some of the tools that we've discussed over the course of these 2 days are the ones that I see being used. Now we're going to have another little panel come up, and I'm willing to be a part of it and a target of it. So we have myself, Patrick, and Raj. I already had mine.
So I was asked to comment on this second bullet. How does the point of departure for risk assessment, how is it determined, and when we're using single cell or single molecule endpoints? And I had some bullets that I sent you, Jessica. I think this is the easy part. The hard part is part one, in my mind, defining the defining the mode of action, because if you have a very good understanding of the mode of action, then as Raj described yesterday, you can easily model it in Simulo, and you have a model of the dose response that can come directly out of that, and from that you can, depending on your decision context, whether you're doing kind of screening level, whether you're doing kind of emergency response where you have to make a decision about, nasty water getting into homes in Houston, or ultimately making a decision that is going to, stand for decades by EPA or another organization. That's enough. Having that model is the basis for making that decision. How we've done this historically is there's a lot of ways, and a lot of them have been adapted for transcriptomics and other new technologies, and I have no doubt that they could be readily used for almost anything that we've discussed over the last day and a half. A lot of times, in vivo assays are simply summarized as the lowest concentration that produced a response. You can do model-based kind of smoothing of that concentration response, and use a process called benchmark dose analysis. It's a little bit it's better, I would say. There's basically, as we've talked about it, understanding that point of departure is an old problem, given that you can define adversity, define your mode of action suitably. So, I put this list of outstanding questions here, and a lot of these we've talked a lot about over the past hour, and I just want to kind of add a little bit to some of these. That one thing that, as we're trying to use these tools, use these models, as the methods get more precise, as we can measure things at a smaller scale, smaller time scale, smaller concentration scale, just because we can measure something doesn't mean that it's useful in a risk assessment context. It doesn't mean that what you're measuring is tied to adversity. Cells have a tremendous capability to buffer against outside influence, and it's on us to define what's the difference between a change at the cellular level that is adaptive, that is just a cell doing its thing, just part of normal biology. If you ate one of those donuts, you're going to have a lot of changes in your body. Decoupling those and there's different ways to define this, whether they're transient, whether they're permanent changes. The adaptive things you can measure from the adverse is outstanding, and I think it becomes more important as we can measure more things with more precision, because there is a kind of a knee-jerk that says if a chemical is causing a change, then that's bad. But in reality, that might not be tied to any adverse outcome. We talked a lot in the last hour, and then also over the last day and a half about how to define normal. What is normal? I would add to that. We have methods for defining that. Traditionally, in frameworks like benchmark dose analysis, it's defined statistically, right? You're looking at a standard deviation in your control animals, or there's different suitable metrics, but as we understand the mechanism more and more, and understand the role of some of these outliers and the different cell populations, maybe that provides a better a better framework for defining what's normal. I think that's a place where I think it's intuitive, but this is a place in the application of risk assessment tools where I can see a direct impact of the kinds of things that we've discussed here. We've talked a little bit about kind of the difference between single cell and bulk response. Is there information that could help characterize risk across population, across demographic groups, gender, age differences? Tailoring chemical safety decisions toward subpopulations of individuals, susceptible subpopulations, whether that's a demographic group, whether it's babies, the elderly, is hot right now, and it's right now we do that in a very crude way. The default is just divide the acceptable dose by 10, and that's your level. But I could see ways that we could use populations of cells using sequencing, using the imaging technologies, genetically diverse populations to understand better what is the variability across these different cohorts, and use that as the basis of decisions. And then my last question is how do we validate this ultimately? And my soapbox is that the standard we're going to be held to is animal studies, and they are not very reproducible in themselves. So two people do the same study, you're looking at maybe a 60 to 80% chance you're going to get the same answer. Michael talked about BPA yesterday a bit. There's been a ton of studies using in vivo studies using BPA. Their points of departure vary from, I wrote it down because I had it handy, between 2 and 1,000 milligrams per kilogram per day. So that's 3 orders of magnitude in the estimate of the concentration that causes an effect in an organism. So that's my caveat when we think about how we're communicating these technologies and their utility. That's the backdrop we should put it up against.
Oh, can you bring up the?
Yeah, one.
Can you?
Yeah, I just want to make sure that the question. Yeah, I have. I just want. Yeah. So I was asked to speak a bit about the last one, which is about does the cell type matter, and if so, these tools will allow us to identify susceptible cell populations, and if so, are the implications different? I would make a slight shift in that terminology there from cell type to a cell state a bit, which better relates to what we talked about yesterday and today. And there are 2 ways to look at that question. One is, yes, of course, we can find the rare cell types, and so we can find the modes of action in a particular way in those cell types and identify why they might be important, and these tools would definitely help us, and we have seen many examples of that. But there's a second issue that I think it's valuable to think about, which is it's starting to, and this relates to what Mel brought up with respect to nonlinearity and thresholding and dose response, and in terms of risk assessment. As we think about the cell states, which of these big clouds for each cell type that we have seen, which ones actually respond to at different levels of exposure, if you will? Which part of that cloud is able to mount an appropriate response to adaptive response or adverse response to at a given level of exposure? And that might be shifting. And so suddenly we are being asked a question, not just cell states, but cell substates within a cloud that might be susceptible in distinct ways, even if they all are pooled as one subtype from a transcriptomics or a proteomic or metabolomic perspective. So how that variability propagates towards a phenotype through the nonlinearities and thresholding and feedbacks is what we would have to end up considering, which means even as we build a complex looking or higher resolution clouds of cell types and a beautiful atlas, without understanding the implications through the nonlinearity and the networks, they just remain as clouds, and it remains cloudy. And so we really have to get at which parts of that gradients of various things that we have seen lead to susceptibility in a meaningful way. And in that context, I want to relate it very briefly to, we talked about what is normal. I think there was also a question of what is healthy also came up yesterday too. And I was thinking about everything that was said, and I don't think of normal, healthy as a state. And I would offer a slightly different perspective from a control engineering sense. And I try to write it down so that I can be a bit clear about this, if possible at all, which is the healthy state, or I would offer it's a trajectory, is the ability of a biological system to mount an appropriate adaptive response to exposure to minimize shift in a phenotype trajectory. So in a control system's point of view, we have to think of what is a reference trajectory? What track are you on? And when a disturbance comes in, are you able to cancel it out in a meaningful way, in a defined way, within a certain time frame, within a certain range, whatever that is? So if you think of it from that perspective, there is no normal state to characterize, really. There is an understanding of what a normal trajectory is as an individual ages, as they go through circadian rhythms during the day, as they sleep, wake up, as they interact in a society, and what have you, and they eat donuts and whatever else, right? So off of the trajectory, what does a perturbation of an exposure do? And there will be many differentials. Some of them are adaptive, as we talked about, at different scales, but some of them would lead to handling of the exposure or disturbance, if you will, over a certain time frame. And so we have to really think about health from that trajectory and ability for a resilient system to mount a response within some time frame. So maybe there's a huge change early on that can be tolerated, high fever as an example, but then you're fine later. So is that a healthy? I would argue it is, even if you were way deviate from a trajectory initially. So I would stop there and offer comments.
Yeah, I'm not sure if I have a question as opposed to a comment, but Patrick, you kind of raised an interesting thing. As a risk assessor, you said, boy, that first part, the mode of action or even maybe mechanism of action, that's the tough part. And the whole time over the last day and a half, I've been thinking that the techniques that we're talking about are really useful for helping us understand the mechanism of action, the mode of action. But God, how do we apply that to risk assessment? And partly because, as we've seen these presentations over the last day and a half, we realize the tremendous heterogeneity within these cell populations. And Raj, it kind of gets back to what you were saying, is, yeah, we can see changes, but ultimately, how do we first define whether that's an adverse effect? And if we know mode of action or mechanism of action, then we can make that call. But then it kind of gets to, well, how many of those cells in that cloud have to undergo that change before we say, well, this could be a problem? And that gets to about what you've been talking about, this dose response. And so I'm having a really hard time envisioning how ultimately we're going to be able to make that call for risk assessment. And again, the point that Raj made is that these aren't just changes, but the kinetics of it is very important. So, for cancer endpoints, maybe, we could make that call more easily. It was like, well, geez, a few cells change, we're really concerned about it. But for non-cancer endpoints, okay, so we see a change that's not leading to cell death necessarily. Okay, so what? We see, a few cells change. But so that's the problem I'm really having in terms of understanding how we use this technology for risk assessment. I certainly understand how we use it to understand mechanism. I mean, these changes are these advances in technology and how we approach toxicology. I think these technologies are very useful, but for risk assessment, and that's probably because I'm not a risk assessor. That's why I'm having a really hard time.
I think that it's going to end up being on us to help define how those changes in cell populations tie to adversity, right? So, I mean, not only do cells have an ability to buffer, but we do too, right? A couple cells, they're gone, right? It's and this might be unfair since the government folks that are here aren't speaking on behalf of their organizations, but my guess is that we will end up taking a very conservative approach, and it will be on the scientists and on the regulated community to establish whether that is appropriate or not. So that's my prognosis.
I'm just reminding you that at least some of the federal folks here aren't speaking on behalf of their organizations. But I think perhaps one way to think about using these data towards risk assessment is to think about what I sometimes think of as the three components of risk assessment. The one is the data, and whether you're going to use cell data or animal data or human data, whatever data you have, and how consistent is it. The models and extrapolations, because if we start with the cell, we've got to take that model somehow from cell to tissue to organ to human being to population. And for those of us who've done extrapolations, if you try and propagate variability through that level of modeling, you're going to get a range of values that are at least 5 orders of magnitude, and the reality is somewhere in there, which isn't very useful for most decision making. And then, the consequences of the right or wrong decision. So what I think, at least in my opinion, from being a risk assessor for a few decades now, these data on the cellular level, including the variability and everything else that we can gather in the systems and feedback loops and modes of action and mechanisms of action, if we can get down there, can help inform what sorts of models, threshold, non-threshold, toxicokinetics, toxicodynamics, what are the important issues to put in our models so our models are simple enough that we can use real data instead of lots of assumptions, as Mel mentioned, or lots of parameters that we have to extrapolate from rodents to people or whatever, and structure different sorts of risk assessment models that seem to comport with the whole range of data we have at the various levels, cellular to population, that makes sense in terms of trying to come up with a pragmatic solution that we, to the extent we can, we validate. Often we can't because we don't want to let people get ill enough to figure out that the model did or didn't work. But without being too conservative, so we can actually do things. Because as everyone in this room also knows, you eat a meal and everything changes, even if it's the healthiest meal you can think of. So part of what I'm thinking of is how can what we learn at the cellular level perhaps help with individual disease interventions, but more importantly, have us think about different ways of taking the data we have, whether it's from laboratory animals or from human populations, and do a better analysis of what might be going on so we can do a better analysis of where the breakpoints are for various levels of exposure and disease outcomes. Knowing that, and this is the hardest part, but I feel we have to keep reminding people that occasionally we're going to be wrong, and we hope not to be, but occasionally we may be very wrong. Because thalidomide is a good example right there. You do the best you can with the information you have and the models you have at the time you make the decision.
I'd like to make a comment. I'm trying to put it in perspective. Risk assessment is a really broad field. Lots of things have to come into making, at the end, a societal decision. It's not just about the scientific information that we can glean by doing these advanced studies. From my perspective, their value is to actually ask, what do we know about biology and how we can kind of organize that to understand processes that occur. This is almost at just a scientific level, and have that information come in and start to inform but not dictate how the field thinks about things like dose response. And then by putting it into a more, into a system that has clear portions, talk about what kind of information would help with the decision making. And also bring tools or discuss tools that this research could help if you're in a laboratory. I mean, one question I would have for you, Norb, is you teach toxicology and how it gets used. How would you see these tools being moved, or some of these tools and the discussion of how they would be moved into a curriculum? I mean, you don't have to answer that, but I think it's a challenge for any of us who have spent time teaching to ask how new material comes in, changes our vision, our view of the world, and then what do you want a student to understand by the time they're done? Because some of them will eventually become risk assessors, and how do you want them to be prepared? And this new information is part of that preparation. How do you collect it? How do you interpret it wisely, correctly? And what do you learn from it? And how does it help with larger decisions?
I would like to add. So the comment about the simplicity of the models, that's not as complicated as a cloud that we have been showing for the last day and a half, I think is very well taken. I want to add a nuance to that, which is that in many times when we're dealing with, it turns out that when we're dealing with all these complex systems, the model might be high dimensional and complicated with many parameters, but the simplicity might come from the number of free parameters. So if you have to think of it slightly differently, for example, I offer an example of whole cell models, where we're talking about modeling everything in a cell, right? And yet, the number of free parameters are surprisingly very few, because all of them have to be somehow linked up and bounded and related in a ratiometric way to achieve a certain cell replication rate. And that puts such a hard constraint on everything else that the uncertainties just don't keep propagating and expanding in that system in parameters. So it may be that as we think about simpler models, we have to be, it's not about having a complex model per se, but having a complex model with few handles that are degrees of freedom is probably how we should think about.
I can agree with that, but I would throw on one thing, which is each of those fixed interactive parameters, and I agree they're interactive, has variability and uncertainty. And if you have 20 such parameters and you put one fixed value, I think there is, I don't remember the author or even the journal, I'm sorry, because I didn't think this would come up today or I would have brought it, but there was an article on epidemiology recently in modeling propagation of disease that was only about the uncertainties in the model parameters that were assumed to be a fixed point, as opposed to a distribution or anything else. And it turned out that for the most part, that was what was driving the modeling and the answers, not the data you were putting in. And I've found this in other modeling that I've done, that if you're not real careful, if you're assuming especially fixed points for known values or known interactions, that especially with limited data, you can have the model providing the answer rather than the data providing the answer. And I have, if you want to discuss it later, I can show you one example that's real quick on that. So it's not that we have to keep the models very simple, but we have to be careful that we're not modeling the model instead of the data.
I think that's a very fair point. Ravi Iyengar calls it, how much did you bake it into your model? The answers that you're looking for versus letting your data. So that the model should have at some level ability to capture a variety of scenarios rather than have an extremely smaller range of what it can predict. At the same time, what you bring up with respect to uncertainty propagation, these are very important. I want to put in a short plug for this committee that was formed out of a multi-scale modeling consortium activities that across agencies actually, called Credible Practice of Modeling and Simulation. That's with focus on healthcare and biomedical sciences, but the rules, we have what are called 10 simple rules along the lines of 10 simple rules for various XYZ, and thinking about how the data influences the modeling choices and the assumptions that go into it, and a third-party independent review, and having competing models. And so there are a whole battery of systems that we recommend for not model credibility, but credible practice of the modeling process itself. And so if you search for CPMS, you'll probably find it in Google. I've just put a plug in. Much thought has gone into it in how to deal with those kinds of issues.
Richard Beeger, FDA. As a chemist/physicist, I've found the last few days of learning a ton of biology very interesting. My first questions is Pat and Raj, you brought up the ideas of dynamics, kinetics. It's really a dose time response. So how many time points are we going to have to start looking at to answer some of these questions? Because obviously what you're talking about is these states. And the states are going to, the initial states, intermediate states, and the final states are going to play a role in some of these responses. And then my second question is the elephant in the room that I know about is the gut micro. In precision medicine, what we've heard about many times in this meeting here, genetics comes out and can now explain maybe 20 to 40% of a patient's response to a drug. How much do you think the cell types are going to play a role in this, given to the fact that we're also missing the gut microflora, which is more cell types than any of these organs that we have right here? So I know that there's two disparate questions here, but I wanted to throw them out there.
I don't have an answer to either one. But I will remark that there are single cell technologies, and Julie Bettine, she just left, highlighted one of these. I mean, the meeting was focused primarily on kind of the sequencing-based modalities, but those non-destructive, where you can observe a cell over time, right? That's the ideal way in my mind to get at those questions of dynamics and look at how you change state. It's just that as far as I know, there's not a way to do that with the transcriptomics. I guess eat fish or something like that. But yeah, I mean there are ways to get at those dynamic questions.
So I might offer that for the dose time response question that you posed, which is a fascinating question. But rather than think about what are the time series that are required to characterize a scheme, a model, a dynamic model, a kinetics very well, I would urge that maybe we ought to be thinking about what are the time points you need to discriminate along multiple options that you might be considering, and that's good enough. So that changes the dimensionality of what you need, because many times the minute you're trying to talk about system identification of nonlinear systems, the number of time points just goes out of control. Whereas if you were to say, what do I need to measure when in order to be able to discriminate on possibilities, then I think that makes it much more feasible and practical in many cases. So we should think about it in that context. And the cell types genetics question that you pointed out, again, I think it comes down to within that cell state, are there cells where the genetics play a role again, as opposed to other substates where that may not be the controlling factor. Brings it down again to what are the controlling points that have an out of proportion effect on the outcome in some way. So that's how I would formulate that problem.
One of the things when we talk about risk assessment is that many of us go back and say, here's a way to do risk assessment. Do the animal study, get the result, apply it, get a number. Now we've got a new technique. Are we thinking, apply the new technique, look at the results, interpret and get a number? I would say right now we're asking ourselves, how in the world would we use these new techniques to collect information on perturbations? We don't know. It depends on what time we would look at it. It depends on what we'll be studying. Quick adaptive responses or longer term responses that require multi-day. We don't know how to do that. And it will differ. One of the answers we got this morning was the answer I told my students is always correct. It depends. It depends on what we're studying. From my perspective, moving forward with these tools in risk assessment is really like developing case law. We have to take some examples and apply them. We have to have work done on the immunological cells, on the testicular cells, and say, here's the problem that we've been studying to understand adversity. Here are the tools, the new tools we can use, and when we apply them, how do we see the world differently? And I would say that it's premature right now to ask how different the world may look. I mean, there are some things we can already say. I would say for me, the world of low-dose extrapolation is completely different because of these tools and technologies, looking at what single cells do and the requirement in most responses to activate a network before you can move to a response. That's a critical piece that has to be discussed. The way I think it has to get brought into the training is to have these cases develop that can be discussed, and then ask the next generation of people to say, how does this make the field look at things differently than we've looked at them in the past? And we don't know. I can't tell you what it will look like. None of us quite know what it's going to look like. And some of us may never see what it looks like. But I'm hopeful that as these technologies get implemented so folks who have had a career in risk assessment can take a look and say, how does it change the way I look at this, and then how does that influence the discussions in the field? But they seem to be transformation. These tools are transformational for how I look at the problems I've dealt with in my career. Other comments? Do we have any online comments? No?